Garry did science the way I like to see it done. When a teacher says that, they say you can see in your student that he took things you told him, expanded them, changed them, and built his own edifice out of things that you can recognize, but he's turned it into something that is characteristic of his lab and not our lab.
From a very early age, I was interested in science. I mean, my parents somehow recognized it. And I got interested in plants, and then that eventually led for me to go to Cornell University, to the School of Agriculture, where I had intended to be a botanist, but very quickly got interested in microbiology. My senior exam was on the T-cell receptor. And then when I came to grad school at Stanford in the fall, Mark Davis had cloned it. And so I found all of that remarkable. Because of the immunology course that I'd taken as a senior, I rotated in the Herzenberg laboratory. I became fascinated by the complexity of the immune system.
Garry was great. I remember him when he first came into the lab, and he was such a character. He was great.
Len and Lee's laboratory were really amongst the first of the open source laboratories. Len insisted that any reagents, such as monoclonal antibodies or what have you, that were developed in the lab be given freely, no questions asked. And it was from the Herzenbergs that I learned a lot of the essentials of how you both develop technology as well as make it useful to the community at large. I saw that algorithms and the way you interpret and analyze data was as important as the collection of the data in the first place.
Garry's had a number of key inventions. The earliest one was probably the retroviral gene transfer stuff, the Phoenix cells. The next big phase, and I'd say this is the invention that really caught my attention when I was not at Stanford, was the single-cell biochemistry stuff. Using a conventional flow cytometer and conventional reagents to look at regulatory cell pathways and cell behavior, and putting them all together to allow you to measure these individual, things in millions of cells per experiment. With that in mind, Garry was always working towards this parameterization. I came in right when he was starting to explore this dark matter, how do I get more parameters so I can get more data points at the same time in a single experiment? Then there was the tissue imaging technologies, multiplex ion beam imaging, which was essentially a solid phase imaging cytoph assay, or something like Codex, which was analogous data but driven by fluorescence and nucleic acid technology. And then beyond that, Garry's done work with single-cell sequencing assays, with split pool synthesis, and now thinking about the next phase of how do we then take all this data and digest it.
The way spatial biology is going now, it's now, there's thousands of groups that use these types of technologies now. One's developed within Garry's lab or in other labs. And I think it really is going to catalyze big developments, particularly in understanding human immunology and cancer that wouldn't have really been feasible otherwise.
I've always been bothered by lost potential. I, again, learned from the Herzenbergs, who would take very roughly hewn individuals and patiently turn them into great scientists.
He's one of those people, it's not just his individual accomplishments, but it's what he's enabled others to do.
We have an ecosystem now at Stanford. I think I have 8 or 9 of my former postdocs in various departments, all doing some kind of spatial biology or immune system biology.
His lab has published in everything from stem cell biology to developmental biology, cancer biology, immunology, neuroscience, data science, all of the ologies. Garry probably has 50, 60, 70,000 citations.
He's just has a track record over decades of pushing out novel capabilities and new biology in a way that I think is pretty unique even to Stanford.
When I finally came down and saw the lab and joined, From the outside, I kind of thought he's really focused on using this technology to do this thing. And then I got to the lab and I realized he was more like a guy at a craps table in terms of things he was trying, right? He knew where he wanted to go, but he had placed bets all over the table. And there was all kinds of little pots of innovation going on in the lab.
One of the things that I've always had a desire to do is to allow questions that should be asked to remain on the table. That led me, one way or another, to being interested in UAP, unidentified anomalous phenomenon, or better known as UFOs. And the data that I've seen convinces me that there are questions worth answering or asking.
At the time when I was a postdoc in the lab, he was taking trips and meeting up with people who were filming these unexplained, like, glowing orbs you'd hear reported sometimes, but was taking a very, again, scientific approach to it.
I could just as well be gardening. So why not do this? Because it gets me up in the day. Had I stayed in the lane of just being a regular scientist, yeah, I meet a lot of interesting people. But I'm telling you, the people that I meet because of this, out of this world.
Garry is my kind of scientist. He's just very into unconventional ideas, unswayed by dogma.
You need to believe in yourself. Trying to convince people that you're right just by talking is not as good as by doing. And it's not about lording over them. It's about saying this will work. And so I'm more than willing to spend that time because I can see it. And so again, it's trusting yourself and not letting, frankly, in some cases, other people bully you. And so what science affords you is the ability not only to be your own boss, but to teach others how to use that technique as well.